Hello, hello. Welcome back to Ali's MCAT podcast. I'm your host, Ali, a pre medical student currently on his gap year, deep in the trenches of MCAT prep. This podcast is all about helping you navigate your MCAT journey. We're covering a variety of high yield topics in an approachable, easy to digest way. While I'm learning the topic, you are too. Think of me as your study buddy, here to guide you through the concepts while you're driving, working out, or taking a break from your textbooks. Today, we're diving into one of the most fascinating topics of biology, embryogenesis and fetal development. This isn't just a biology chapter, it's the blueprint for life itself. We'll explore how a single cell transforms into a fully developed human being, touching on early developmental stages, cell communication, fetal circulation, and the milestones of gestation and birth. So, grab your notes or just relax, stay safe, and let's explore this incredible journey together. Segment 1: The early stages, from zygote to germ layers. Alright, let's get started with the very beginning. Fertilization. It all begins when a sperm fuses with an egg, forming a zygote. That's the first chapter of life. The zygote undergoes cleavage, a series of rapid cell divisions leading to the morula, a solid ball of cells, and that transforms into a blastula, a hollow structure with a fluid-filled cavity called the blastocell. Picture it like a water balloon ready to attach itself to the uterine wall. The next step is implantation, where the blastula embeds itself into the uterus, which is a vital step for establishing nourishment. Unfortunately, if this process goes awry, as in an ectopic pregnancy, the embryo implants outside of the uterus, posing a risk to the mother. Once implantation occurs, the embryo undergoes gastrulation, forming your 3 germ layers. You'll know them as the ectoderm, mesoderm, and endoderm. So the ectoderm is the outer layer, which forms the nervous system, skin, sensory organs. Think of it as attractoderm, things that make us attractive, like our appearance and our intelligence. The mesoderm is the middle layer, creating muscles, bones, and your circulatory system. Think of it as mesoderm, establishing movement and transport within the body. But finally, your endoderm, the innermost layer, forming the linings of your internal organs, like the digestive and respiratory system. Alright, next we have neurulation, where the neural tube forms, eventually becoming a brain and spinal cord. Kaplan emphasizes the role of folate in this stage, as a deficiency can lead to severe deficits like spina bifida and encephalopathy. Now that the embryo has its layers, how does the cell decide its roles? Enter cell specialization and communication. The first step is called determination, where the cells commit to a specific fate. This is followed by differentiation, where they develop their structure and function for that role. For instance, if a cell is destined to become a neuron, it'll grow axons and dendrites to transmit signals. Stem cells play a starring role here. Totipotent stem cells can become any type of cell. Pluripotent stem cells are slightly more restricted, but they're still incredibly versatile. Multipotent cells are specialized but limited to specific lineages. But how do cells communicate with each other? It's through cell-to-cell communication. So you'll have 4 types of signaling: autocrine, paracrine, juxtacrine, and endocrine. Autocrine signaling is when cells signal themselves, setting like a personal reminder. Paracrine signaling is like a short-distance communication with your neighbors. Juxtacrine signaling is the direct contact-based communication, and endocrine signaling is long-distance communication through the bloodstream. An example of all of this is induction, where one group of cells guides another. During eye development, the optic vesicle influences nearby ectoderm to form the lens play code, a fascinating dance of reciprocal signaling. Alright, now let's talk about fetal circulation, a system designed to bypass the lungs and liver of the baby, which are not yet functional. The placenta acts like a lifeline, exchanging nutrients and oxygen between the mother and the fetus. Umbilical arteries that carry deoxygenated blood from the fetus to the placenta, and there's one umbilical vein that brings oxygenated blood back to the fetus. To bypass inactive organs, the fetus uses three shunts. These shunts are the foramen alveoli, the ductus arteriosus, and the ductus venosus. The foramen alveoli connects the right and left atria, which bypasses the lungs. The ductus arteriosus diverts blood from the pulmonary artery to the aorta. The ductus venosus directs blood from the umbilical vein to the inferior vena cava, which bypasses the liver. This ingenious system ensures that oxygen and nutrients reach vital organs, like the brain. The final act is gestation, which is divided into 3 trimesters. First trimester, organogenesis, or the formation of major organs, begins. In the second semester, the fetus grows rapidly and movement becomes noticeable. In the third trimester, the fetus gains weight and prepares for life outside the womb. Finally, birth, or parturition, occurs in 3 stages: cervical dilation, delivery of the baby, and delivery of the placenta. It's a remarkable process, coordinated by hormones like prostaglandins and oxytocin, which trigger contractions and ensure smooth delivery. And that wraps up our exploration of embryogenesis and fetal development. From a single zygote to the miracle of birth, it's a story of precision, resilience, and collaboration. If you found this episode helpful, be sure to subscribe and share it with your fellow pre-meds. Next time, we'll be diving into other high-yield topics, breaking it down step by step. Until then, happy studying, and let's crush this MCAT together. Bye.